And the question is, activated kaolinite clay is used to manage. So the options given for this are subterranean insects or pests, leaf feeders, those which mainly focuses on feeding the leaf, storage pests, so those pests which occurs only in the storage godown, and stem borer, and also in storage godown as well as in the uh, home or somewhere, and stem borer. And answer is not no. What is activated kaolinite clay? It is a processed kaolin clay, and also we can call it as China clay. Okay, so chemically it is basically a hydrated aluminate silica. Okay, activated means that it's been heat treated or acid washed one to increase the surface area and absorbing capacity of the clay. Okay, how the clay usually kills the insect? So through desiccation, abrasion, and interference. What is desiccation? That and all right? Desiccation means like it will lose the water. The insect will lose the water. How? This fine particle, this clay will have a huge surface area, right? So when it contacts with the insect cuticle, this will be having the adsorption on the cuticle, because the insect layer is having waxy layer, right? So in which it will be absorbed. So this destroy the waterproofing barrier of the insect. So wax layer is the waterproofing barrier for the insect, right? So since it absorbs on the particular layer, it breaks the waterproof barrier, so that insect will lose their water, and it will die because of the desiccation. And abrasion is also one of the reason. What is abrasion? The sharp edge of this particular particle abrade the cuticle, so that water losses will occur. Okay, and also it blocks the spiracles and all, and so that it prevents the respiration and feeding as well. Okay, and the correct answer is storage space. So this activated kaolinate clay is used as a inert dust grain protectant. Okay, in the storage structures and all. Mainly it affects the rice weevil, lesser grain borer and all. Okay, and pulse beetle as well. How it is commonly used means we can mix 1 to 2% with the grains. Okay, before storage. Okay, in the floors or bags as well. It's safe for the human consumption at these doses, and it is also approved in the organic storage. Okay, we have to mix 1 to 2% with the grains. And why others are wrong means that subterranean pests usually they live in the soil and they have a moisture around them, right? So kaolin dust loses their efficacy when it was wet, and doesn't persist with the soil pest, so that it won't come. So for the subterranean pest and all, we usually recommend chlorpyrifos and all. Okay, physical dust is not commonly used. And leaf feeders again, the unprocessed kaolin is used as a particle film in the orchards, but activated kaolinate is generally referring to the processed dust. So field application of this activated clay is rare, because rain, if the rain comes, it will wash off. Okay, so it is totally uneconomical, unsuitable for the leaf feeders. Stem borers, same. These pests generally live inside the plant stems and all. So the contact dust like this kaolin cannot reach them. So the control requires the systemic insecticides, not the dust particle application. So physical dust is not effective one against internal feeders and all. Okay. And the next question is the tip clipping of infested seedlings to manage rice stem borer, Sitpopaga insectulus, or in the rices. So there is one practice usually followed in the rice field, that is tip clipping. So the seedling tip will be clipped. Okay, so that way, which is the control method they asked, whether it comes under the mechanical, physical, or cultural, biological. They asked, what is tip sleeping? So generally in nurseries and all, no, rice nurseries will be there. If stem borer eggs or egg larvae are present, farmers usually clip 2 to 3 centimeter of the seedling tip before transplanting, because the seedling tip where the stem borers usually lay the eggs, right? So before they go for a transplantation, they usually clip it off. Okay, so that they will able to destroy the life cycle of the particular pest yellow stem borer. So the main reason is that stem borer moth lay the eggs on the leaf tip. So the eggs and early larvae are removed with the clipped portion. So this will directly reduce the pest population going into the main field. So the options, different options are given. So you may be knowing this tip clipping means we are doing mechanically, right? So the correct answer is mechanical control, which is the physical removal or killing of the pest using manual labor or tools or something. Okay, so you physically remove the infested plant with your hand or tool. So no chemical is applied and no living organism is used and no change in agronomic practices is usually done. So it is a direct and mechanical method. Okay, and physical is usually, this control uses the physical forces like heat, cold, light, sound, radiations, and all. If you are using some energy means, then we can tell that will come under the physical control. For controlling flies and all, we will be installing the light, no, some light, keto light and all, it will be there for flies and all. So that in that case, light will be used. So that is the physical control method. For example, hot water treatment, light trap and sticky trap, mulching and all come under the physical control. Cultural means we are modifying the agronomic practices. So that is no more related with the this mechanical control. And biological, where we commonly use living organisms to control the pest. Okay, so the correct answer is mechanical control. The next question is, in honeybees, which enzyme usually converts sucrose into dextrose and levulose? So generally what happens in honeybees means, honeybees generally collect the nectar, right? Which is mostly sucrose. So the main component of the nectar is sucrose. Okay. So in the honey stomach or, honeybee hope, where bees will add the particular enzyme to convert the sucrose into simple sugars like dextrose and levulose. Dextrose means nothing but glucose. Levulose means fructose. Okay, so they are adding the particular enzyme along with the sucrose so that the complex sugar will be converted into the simple sugar. Why they are doing like that means, so that they will able to reduce the water content present in the honey, and also it helps in preventing the crystallization and makes the honey easier to digest and store and all, and it will give the honey extra sweet. Okay, so that's why. So the options given for this are nuclease, invertase, amylase, sucrase, and the answer is not known. And the correct answer is invertase. Okay, this is a standard term used for honeybees. Okay, so this will hydrolyze the sucrose into the equal parts of glucose and fructose. Okay, so this is also called invert sugar. Invert sugar means sucrose that's been inverted into dextrose and levulose. So this name comes from the change in the optical rotation during the hydrolysis. Okay, so this is the main enzymes. So this enzyme is present in their salivary glands and all. Okay. And nuclease is generally break down the nucleic acid, DNA, RNA, and all. So the digestion of nucleic acid in the food, it is not helpful for the conversion of sugars. Okay, this is having no role in the honey formation. And amylase and all, this will help for the breaking down the starch into maltose and dextrin. Okay, even the honeybees secrete the amylase, but it acts on the starch and dextrose, not on the sucrose and all. Okay. And sucrase is chemically the same as the invertase, but it hydrolyzes the sucrose. In mammals and humans and all, the enzyme in the intestine is called sucrase. So in the honeybees, we usually call it as invertase. Okay. So in insects and the honeybees and all, the same enzyme. Sucrase is the invertase over there. So that's why. So invertase in honeybees will make the sucrose into glucose and fructose, and amylase will make the starch into maltose and dextrine. Okay. And the next question is, which of the egg parasitoid was employed for the control of sugarcane shoot borer? Okay. So to control this particular pest, there is a usage of the biocontrol agent. Which one they asked? That is the egg parasitoid they determined. What is egg parasitoid? So those parasitoid adult will able to lay their eggs in the body of the pest. Okay. And once the egg deposition is done, the larva will emerge out, and this will, feed the pest so that it will get killed. Right? So sugarcane shoot borer here, what they mentioned is that sugarcane yearly shoot borer and all. Infus catalyst. This is the major and yearly season pest of sugarcane in India. And here this larva usually bores into the shoot and causing the dead heart. So for its management, egg parasitoids are commonly used as the key component of the integrated pest management. Okay, so there is a one parasitoid which is widely used in the India. They have given. We have to choose them. So the options are Trichogramma japonicum, Trichogramma australicum, Trichogramma kylonis, Trichoderma viridae, and the answer is not known. And the correct answer is Trichogramma kylonis, which is the egg parasitoid which lays the eggs inside the egg of the kylo infus catalyst, right? So egg parasitoid means during the egg stage of the particular borer, it will lay the eggs. Okay, so that the developing parasitoid kills the host egg. So remember one thing, egg parasitoid means that the particular adult of this parasitoid will lay the eggs on the egg of the pest. So before the larva emergence, before the particular pest larva emergence, the parasitoid, will be, able to kill the pest. So the developing parasite kills the host egg. So this will prevent the larva from hatching. Okay. So it is mass produced and released in the sugarcane field. This Trichogramma kylonis is mass produced and it is released in the field around 1 lakh and 1 lakh adults and all, it can be released per hectare. And it usually be released at different time intervals. 6 to 8 releases can be done at weekly intervals starting from 45 days after planting and all. Okay. And Japonicum is mainly used for the rice stem borer and all, not the primary choice for the shoot borer. And also Australicum can be used for the sugarcane borers, but in India, Calonis is common and standard one. And the Trichoderma viridae is the fungus, not an insect parasitoid. It is a biocontrol agent against the soil-borne plant pathogen like Rhizoctonia, Fusarium and all. And the thing is that since it is similar to the Trigogramma, it is totally different organism. So it is like a trap for you. Okay, you have to be sure on this. Trigogramma kylonis. And the effective larval parasitoid to control Osceola oryzae in rice ecosystem. What is larval parasitoid means? The particular adult parasitoid will lay the eggs on the larval stage of the host, right? So that is larval parasitoid. So to control the Osceola oryza, which is nothing but the gall mitch, right? So here the larva of this particular pest usually feeds inside the rice plant and causes the silver shoot and onion leaf and all. So this is the major pest in the lowland tribes. And the different options given for this are Dracon brevicornis, Trichogramma calonis, Platygaster oryzae, Trichospilus pupivora, and an answer is not known. So the answer is Platygaster oryzae, which is the larval pupal endoparasitide of rice gallmilch. Okay, which means that it lays the eggs during the larval stage of the Osceola oryza, and during its nymphal stage it will come out as an adult. Okay, this is a larval pupal endoparasitoid of rice gallmilch, which is the Osceola oryzae. So the female parasitoid lay the eggs inside the gallmilch larva, and the parasitoid larva develops inside and kills the host before it pupates. Okay, and kill it, and it will come out as the adult. It is host specific to the Osceola oryzae, and it is a key natural enemy used in biological control in India. Platygaster oryzae and all. So Bracon brevicornis, it is a larval ectoparasitoid. Ecto means it will lay the eggs on the larva, not inside. It attacks most of the Lepidopteran larvae like Coxera, Floria, and Spodoptera and all, not used for the gallmilch. Gallmilch is the usually dipteran one. Okay, gallmilch is the fly. So this Bracon brevicornis doesn't target the fly. And Trichogramma and all, usually it is the egg parasite. It parasitizes the eggs of the moth, like sugarcane shoot borer, and cotton bollworm, and rice tumbler and all. Okay, not for the Osceola oryzae. And the Trichospilus pupivara, it is a pupal parasite used for the Lepidopteran pest for the coconut black-headed caterpillar. Okay, Opicina arnosula, not for the Osceola oryzae. And the next question is, while taking stored grain samples to decide the need for the shed fumigation, the infestation level is categorized as mild if the number of insects per kg and the sample is. So remember one thing, if the number of insects, there should be some number, right, per kg. Then only it is categorized whether it is a mild, moderate or severe. So here they already categorized that is a mild, and they, those will be needing for the shed fumigation. Okay, and they ask like how many numbers should be present per kg. Option A nil, up to 2, up to 5, up to 10, and answer is not known. And what is shed fumigation means? Fumigating the whole godown or shed or warehouse along with the grain stored inside. So stored grain will be there inside, and totally they will fumigate. Instead of fumigating the individual bags and all, they will fumigate the whole godown. It is one of the standard method used by the FCA and all state warehousing corporations and all, they will use. Okay, how it is usually done means first they will seal. So the shed or godown is made airtight by sealing doors, windows, ventilators, and cracks with the mud, tape, and polythene. So completely the entire godown will be sealed. Okay, and they will choose whether they go for the tablets or some gases and all. So mostly they will choose the fumigant pellets like aluminum phosphate, that is a tablet that will be placed on the floor or in between the bags and all as per the dose. Then there will be some exposure period. The shed will be kept closed for 5 to 7 days. So the gas will be diffused completely and kills all the stage of insect, right? Whichever is present inside the grains, bags, and structures and all. So after the particular days, what they do means the shed will be opened again and ventilated for 2 days before handling the grain. Okay, so that's the reason, and how this is how it is being done. And the correct answer is up to 2. So if the insects are up to 2 per kg means that is considered as the mild one. Okay, and 0 means nil. 3 to 5 means that will be considered as the moderate one. If there is a more than 5 insects present per kg of grain means that will be the severe one. Okay, so the option is up to 2 only. Okay, up to 5 means that is a moderate, and more than like up to 10 means that is a severe. Okay, and the next question is the storage pest, which is also used for the mass production of trichogramma. So what is trichogramma? This is the genus of egg parasitoid wasp, right? So this female of this particular wasp will lay the eggs inside the eggs of the host insect. The host means our targeted or pest insect, so that it will kill before the pest can hatch. So it is one of the most widely used biological control agent for most of the Lepidopteran pest, right? So why the mass production is required means you need a millions of trichogramma adults, right? Just for the field release, but they are too small. They are too small means the size is a half centimeter, like 0.5 mm will be there. The adult of this particular egg parasite size is very small, 0.5 mm. They are too small, and we have to rear, right? So we need more, because for sugarcane, borers and all, if we are releasing means we need 1 lakh adults, 1 lakh numbers of adults can be released per hectare at weekly interval. Okay, 4 to 6 release or 6 to 8 release can be sometime done. Okay, it depends on the how our pest population will get reduced. So in order to multiply it, we need the mass production, right? So that's why we are rearing it in the lab. Okay, and generally the pest, which pest is commonly used for the mass production of this particular insect, because this will lay the eggs on the particular eggs of the pest, right? So that from the eggs of the host, the adult of this parasite will come out, and we can collect it and we can release it. Okay, so the options given for this are Coxera cephalonica, Cytotraga cerealia, Ephistia cautela, Plodia interpunctuala, and the answer is not known. There is Coxera cephalonica, which is the rice moth. So this is the stored grain rice moth. It's very easy to culture on sorghum, maize, and all in the lab. Okay, so it lays the thousands of eggs, which are large and UV treatable. Right. So the trichogramma is readily parasitize the Coxera eggs. So this is the perfect host for the mass production of this particular parasitoid. Cytotraga cerealia and all, it is an Anguma's moth. It can be used in some countries, but not the standard one in the India. Ephistia cautela and Plodia interpunctuala is used occasionally in the research, but not for the mass multiplication or the large scale production in the India. So Coxera for trichogramma. It's commonly used. The next question is, which of the following is not a juvenile hormone analog? What is juvenile hormone analog? It is a synthetic chemical, okay, which usually mimics the juvenile hormone in insects. What is juvenile hormone? So those hormone will keep the insects in the immature larval or an impulse stage, right? So those will be occur during the immature stage, juvenile hormone. Okay, so analog means it is a mimicking the juvenile hormone. So this juvenile hormone analog will disrupt the metamorphosis. Metamorphosis means life stages of insect, right? So the insects cannot mold to adult. It will keep the insect in the, or it will keep the insect remained in the larval or pupal stage. It cannot mold to adult properly, so that it will die. So they are considered as the insect growth regulator also. It is used in the stored grains and all. So the options given for this are actually juvenile hormone analogs are there, but among them one is not the juvenile hormone analog. That is that which one we have to find. Okay. The options are hydroprene, chloropicrin, kinoprene, metoprin, and the answer is not known. And the answer is chloropicrin. So this is not a juvenile hormone analog. This is basically a fumigant, or we can tell the soil sterilant, not a hormone mimic. Okay. And this will act as a respiratory poison used to control the soil insects. Okay, and it was even used as a tear gas, right? But it has nothing to do with the juvenile hormone. And others are hydroprene, kynoprene, methoprene, and all. They are the juvenile hormone analogs. Okay, and the brand name will be different. If it is ending with prene, prene means those are juvenile hormone analog. In that way, you can easily remember. The next question is, which of the crystal protein produced by the soil bacterium Bacillus thuringiensis effective for the coleoptera pest? So you may be knowing about the BT crops, right? So in those crop, this particular gene will be, injected. So that is one of the, transgenic crop. So they asked which crystal protein of the bacteria is effective against the, coleoptera pest. And the options are Cry1, Cry2, that is a crystal protein, right? And Cry3, Cry4, and the option is not known. Answer is not known. So the correct answer is Cry3. What is Cry proteins means that BT generally produces the crystal proteins, right, during sporulation. And this is called Cry proteins, or else we can tell endotoxin. Okay, delta endotoxin. Delta endotoxin. So when the insect is ingesting this particular toxin, they bind to the toxin will bind to the midgut, and it will cause the insect death. So here the options are different. The correct option, which is opt for coleoptera control, is Cry3. Cry1 is mostly for Lepidopteran control. Okay, and Cry2 is Lepidopteran as well as Dipteran. Cry3 is meant for the coleoptera control. Okay, Cry3 means coleoptera, and Cry4 is again for Dipteran. Okay, and not for the coleoptera. So the correct answer is Cry3 protein. Target insect pest of transgenic BT cotton or dash in India. So this is the question. The options given for this are bollworms plus Prodinia, which is the Noctuday moth. Like we can tell this Spodoptera, right? And Prodinia plus boll weevil, bollworms plus stem weevil, then bollworms plus boll weevil, and finally the answer is not known. Okay. And the correct answer is bollworm plus Prodinia. So generally what BT cotton targets in India is, so basically in India, it expresses the Cry1AC earlier, and later Cry1AC and Cry2AB and all will be formed in the BT cotton. Okay, from the Bacillus thuringiensis. So these toxins will be effective against Lepidopteran insects. You know how it is acting and all. And the main target pest or the bollworms, right? That is American bollworm, Helicoverpa armigera, and pink bollworm, which is Pectinophora gossypiella, spotted bollworm, that is Cereus bitella, and spiny bollworm, that is Cereus insulan and all. And what is Prodinia means that this is the Spodoptera litura. Okay, and also we can call it as tobacco caterpillar or cutworm, common cutworm. So BT cotton was mainly introduced specifically to control these bollworms, which are causing 30 to 60% yield loss. Okay, and also to control the Prodinia. So why other options are wrong means here the boll weevil, which is the Anthonymous grandis, which is the coleoptera, right? So BT cotton, basically, which the earlier where the BT cotton released was not against the boll weevil and all. Okay, and because the earlier the first one, boll god 1 and 2 and all, doesn't have the Cry3 toxin, so it is not effective against weevils. Okay, so it is not suitable. And here the bollworms plus stem weevil. Again, the Pemperellus savinus is a coleoptera pest of the cotton in India. So it is not controlled by the BT cotton. And the bollworms plus boll weevil also. It is also having the same reason as the above. Okay. So BT cotton means the Lepidopteran control only, mainly. If the particular gene Cry3 is there, means that may be effective against coleoptera. So this is how you should remember the transgenic bt cotton target pest. Cry1 AC means cry1, cry2 and all usually effective against lepidopteran pest and all. If the crop is having the cry3 genes, that is a Volgod 3 maybe later, right? So that is effective against coleopteran. Okay.